Very pleasant morning to one and all present here. Today we learn about the topic polymerase chain reaction. Introduction. What is polymerase chain reaction? Polymerase chain reaction is nothing but a technique which is used to make numerous copies of a specific segment of DNA quickly and accurately. The polymerase chain reaction enables investigators to obtain the large quantities of DNA that are required for various experiments and procedures in molecular biology, forensic analysis, evolutionary biology, and medical diagnostics. PCR was developed in 1983 by Carrie B. Mullis, an American biochemist who won the Nobel Prize for Chemistry in 1993 for his invention. Before the development of PCR, the methods used to amplify or generate copies of recombinant DNA fragments were time-consuming and labor-intensive. In contrast, a machine designed to carry out PCR reactions can complete many rounds of replications, producing billions of copies of DNA fragments within few hours. Let's learn about the principle of operating PCR. PCR technique is based on the natural processes a cell which is used to replicate a new DNA strand. Only a few biological ingredients are needed for PCR. The integral component is the template DNA, that is, the DNA that contains a region to be copied, such as a gene. As little as one DNA molecule can serve as a template. The only information needed for this fragment to be replicated is the sequence of two short nucleotides, that is subunits of DNA at the either end of the region of interest. These two short template sequences must be known so that two primers, short stretches of nucleotides that correspond to the template sequences can be synthesized. The primers bind or anneal to the template at their complementary sites and serve as the starting point for copying. The DNA synthesis at one primer is directed towards the other, resulting in replication of the desired intervals of sequences. Also needed are free nucleotides, which is used to build the new strands, and the DNA polymerase, an enzyme that does the building by sequentially adding on free nucleotides according to the instructions of the template. Steps involved in PCR. PCR is a three-step process that's carried out in repeated cycles. The initial step is the denaturation or separation of the two strands of the DNA molecule. This is accomplished by heating the starting material to temperatures of about 95 degrees centigrade. Each strand is a template on which new strand is built. In the second step, the temperature is reduced to about 55 degrees centigrade so that the primers can anneal to the template. The third step, the temperature is raised to about 72 degrees centigrade and the DNA polymerase begins adding nucleotides onto the ends of the annealed primer. At the end of the cycle, which lasts about 5 minutes, the temperature is raised and the process begins again. The number of copies doubles after each cycle and it will go up to 25 to 30 cycles produce a sufficient amount of DNA. The original PCR procedure, one problem was that the DNA polymerase had to be replenished after every cycle because it's not stable at the high temperatures needed for denaturation. This problem was solved in 1987 with the discovery of heat-stable DNA polymerase called TAC, an enzyme isolated from the thermophilic bacterium Thermus aquaticus, which enables hot springs. TAC polymerase also led to the invention of the PCR machine. So this is a comprehensive diagram which shows how two strands of DNA getting replicated or getting amplified to several million folds. Here is your template DNA and primers are added. Tag DNA polymerase, which is a heat stable enzyme, works at 95 degree, is added along with DNTPs like DTTP, DCTP, DATP and DGTP. And here is the template strand now getting separated, widely separated at denaturation step, which is the first step involved in PCR. This takes place at 92 degrees centigrade, and now the primers are getting annealed, which works at 72 degrees centigrade. This temperature is slightly lower. Now to the primer, DNA polymerase enzyme is added and DNTPs are added just complementary to the template strand. by which the daughter strand grows just complementary to the template strand. So at the end of the first cycle, two strands of DNA becoming a fourth strand. So each cycle repeats, that is every cycle repeats, excuse me, and the DNA is getting amplified to several applications. Because DNA from a wide range of sources can be amplified, the technique has been applied to many fields. PCR is used to diagnose these genetic diseases and to detect low levels of viral infection. In forensic medicine, it is used to analyze minute traces of blood and other tissues in order to identify the donor by his genetic fingerprint. The technique has also been used to amplify DNA fragments found in preserved tissues, such as those of 40,000-year-old frozen woolly mammoth or of a 75,000-year-old found in a peat bog. Thank you.